Welcome to the deep dive. Today we're really getting into the nuts and bolts of the male genitourinary system.
That's right. We're focusing specifically on Chapter 42 from Porth's Essentials of Pathophysiology.
Exactly. Our mission here is to break down the core stuff, structure, function, hormones, how things change with age, basically, make it all click for you, like an audio study guide.
And you really need that foundation. We're talking about the testes, the ducts, accessory organs like the prostate, the penis. It's all connected.
Two main jobs, right.
Yeah.
Making hormones and germ cells, and then, getting those sperm where they need to go.
Precisely. And understanding the normal is absolutely key before you can grasp what goes wrong in diseases. The whole story really starts way back. In utero.
Okay, let's go there. Embryonic development. It all hinges on that Y chromosome, doesn't it? Specifically the SRY gene.
Absolutely. If that SRY gene is present and functional, the primitive gonads get the signal, become testes. If it's absent, they develop into ovaries. It's that fundamental switch.
But it's wild that initially, the embryo has the potential for both male and female internal structures.
It really is. For about the first 7 weeks, you've got both wolffian ducts, which are the male precursors, and müllerian ducts, the female precursors, just sitting there.
So, like duplicate plumbing, how does the body sort that out?
Hormones. The newly formed testes start producing 2 critical things almost immediately. First, anti-müllerian hormone, or AMH.
Okay, AMH. Sounds like it does what it says on the tin.
Pretty much. It actively suppresses the Müllerian ducts, causing them to regress. So no uterus, no fallopian tubes in the male.
And the second hormone?
Testosterone. Produced by the Leydig cells in the testes. Testosterone gives the green light to the wolffian ducts.
Ah, so it tells them to develop into the internal male structures.
Exactly. Epididymis, vas deferens, seminal vesicles, that whole internal network.
Okay, but wait. The source material highlights something crucial about external parts, like the penis and prostate. Testosterone alone isn't enough.
Correct. This is a key point. For the external genitalia to form correctly, testosterone needs to be converted into a more potent form, dihydrotestosterone, or DHT.
How does that conversion happen?
There's an enzyme called 5-alpha reductase, mainly in the target tissues, that does the job. DHT is what really drives the development of the urethra, prostate, and scrotum.
So here's the aha moment then. If an XY embryo, genetically male, somehow lacks testosterone or can't convert it to DHT,
then despite the Y chromosome, the external genitalia will develop along female lines. It underscores just how critical those specific hormonal signals are at that specific time. No signal, female pathway proceeds.
Fascinating. Okay, fast forward. The testes are formed, but they end up outside the main body cavity in the scrotum. Why?
Temperature. Pure and simple. Sperm production, spermatogenesis, is incredibly sensitive to heat. It needs to be about 2 to 3 degrees Celsius cooler than the core body temperature.
So the scrotum acts like a cooling unit.
Essentially, yeah. And it has ways to fine-tune that temperature. There's the dartos muscle on the scrotal wall. When it's cold, it contracts, wrinkling the skin and pulling the testes closer to the body for warmth.
And there's another mechanism too, right? Something involving the blood vessels.
Yes, the pampiniform plexus. It's this intricate network of veins wrapped around the testicular artery.
Like a heat exchanger.
Exactly like a heat exchanger. The cooler venous blood absorbs heat from the incoming arterial blood before it reaches the testes. It's remarkably efficient at maintaining that lower temperature.
And clinically, if that descent doesn't happen properly, cryptorchidism.
Big problems. If the testes remain in the abdomen, the higher temperature usually prevents normal sperm production. Plus, the pathway they take, the inguinal canal, needs to close properly after descent. If it doesn't, that creates a weak spot, a risk for inguinal hernias.
Okay, factory-built, temperature-controlled, let's talk production. Spermatogenesis, inside the seminiferous tubules, which are incredibly long.
Unbelievably long. The source mentions if you uncoiled them all, it'd be something like 750 feet. That's where the magic happens, starting around puberty, about age 13.
And lining these tubules are the Sertoli cells. What's their job?
Think of Sertoli cells as the nursemaids and managers. They provide structural support, nourish the developing sperm cells, form the blood-testis barrier, and importantly, they secrete hormones like inhibin and even some estradiol.
Okay, let's walk through the sperm creation process itself. It takes a while, right?
It does, about 90 days from start to finish for a single sperm cell to mature.
And it starts with diploid cells.
Right. The spermatogonia. These are the stem cells. They divide by mitosis to keep their numbers up, but then some enter meiosis.
Meiosis, that's the reduction division.
Exactly. They become primary spermatocytes, then secondary spermatocytes, reducing the chromosome number by half. The result is haploid spermatids.
But spermatids aren't quite sperm yet.
No, they still need to undergo spermiogenesis. That's the final transformation. They streamline, grow a tail, flagellum, develop the acrosome cap needed to penetrate the egg, and become the mature spermatozoon. Ready for action almost.
Almost. Where do they go next?
From the tubules, they squeeze into the rete testis, then through efferent ducts into the epididymis. This structure is crucial.
Why is that?
That's where sperm finish maturing and, critically, gain their motility, the ability to swim. They're not really functional until they've spent time there.
And after the epididymis.
They travel up the vas deferens, also called the ductus deferens. The end portion widens into the ampulla, which is the main storage reservoir for mature sperm.
Ah, and that explains the vasectomy point. Why fertility lingers for weeks after the procedure.
Exactly. You've got to clear out that stored supply in the ampulla and the rest of the duct system. It takes about 4 to 5 weeks usually.
Right. Now, sperm aren't ejaculated alone. They're mixed with fluids from accessory glands to make semen. First up, the seminal vesicles.
Yes, they contribute a significant volume, about 60% of the semen. Their fluid is thick and rich in fructose.
Fructose for energy.
Precisely. Fuel for the sperm's long journey. They also add prostaglandins, which might help stimulate contractions in the female reproductive tract.
Okay, next gland. The prostate sits right below the bladder.
And surrounds the urethra, which is clinically important. The prostate adds a thin, milky fluid. It contains things like citric acid, calcium, acid phosphatase, but the key component is its alkalinity.
Alkalinity, why is that so vital?
Because the environment in the vas deferens is slightly acidic, and the vaginal environment is quite acidic, sperm motility is severely hampered by acid. The prostate fluid neutralizes that acidity.
Bringing the pH up to that optimal range around 6.0 to 6.5.
Exactly. Essential for sperm activation and survival. Without it, fertilization is basically impossible.
And because it wraps around the urethra if the prostate enlarges.
That's benign prostatic hyperplasia, BPH, very common in older men. The overgrown tissue, usually the intermucosal glands, squeezes the urethra, causing urinary obstruction difficulty starting, weak stream, that sort of thing.
Makes sense. And there's one more set of glands.
The tiny bulbourethral glands, or Cowper's glands, they secrete a clear alkaline mucus before ejaculation.
What's the purpose of that?
It helps neutralize any acidic urine residue left in the urethra, kind of paving the way and lubricating the tip of the penis. So you get pH protection at multiple levels.
Okay, we've got the structure, the production, the fluid. Now, how is it all controlled? The hypothalamic pituitary gonadal axis.
Yeah.
Sounds complicated.
It's a classic negative feedback loop, really elegant once you break it down. It starts in the brain, the hypothalamus,
Which releases?
gonadotropin-releasing hormone, GnRH. It travels just a short distance down to the anterior pituitary gland.
And the pituitary responds by releasing 2 different hormones.
Yes, the gonadotropins. Luteinizing hormone, LH, and follicle-stimulating hormone, FSH. These travel through the bloodstream down to the testes.
And they have different jobs once they get there.
They do. LH primarily targets the Leydig cells, those cells located between the seminiferous tubules. LH stimulates them to produce and secrete testosterone.
Okay, LH makes testosterone. What about FSH?
FSH targets the Sertoli cells, the ones inside the tubules that we talked about earlier, the nursemaid cells. FSH is essential for initiating and maintaining spermatogenesis.
So FSH gets sperm production going.
Right. And importantly, FSH stimulation also causes those Sertoli cells to produce another hormone called inhibin.
Inhibin. Okay, so now we have testosterone and inhibin being produced by the testes. How does the feedback work?
It's a dual system. High levels of testosterone circulating in the blood feed back to the hypothalamus and pituitary, telling them to decrease the release of GnRH and particularly LH.
So testosterone controls its own production via LH. Makes sense.
But if that were the only feedback, high testosterone might shut down FSH too much, hindering sperm production. That's where inhibin comes in.
Ah, so inhibin provides separate feedback.
Exactly. Inhibin, produced by the Sertoli cells in response to FSH, specifically feeds back to the anterior pituitary and inhibits the release of FSH.
Clever. So you can regulate testosterone levels and sperm production somewhat independently.
Precisely. It maintains balance for both the systemic effects of testosterone and the ongoing need for spermatogenesis.
Let's talk about those systemic effects. Testosterone does a lot more than just sperm stuff, looking at chart 42 to 1 in the source.
Oh, absolutely. During fetal development, it's crucial for differentiating the male reproductive tract. Then at puberty, it drives the development of primary and secondary sex characteristics, penis and scrotum growth, deepening voice, body hair, thicker skin.
And it has major anabolic effects, right? Building muscle.
Huge anabolic effects. It promotes protein synthesis, leading to significant musculoskeletal growth. The source mentions up to a 50% increase in muscle mass during male puberty compared to females.
That anabolic power explains why some athletes misuse synthetic androgens. The book mentions the downsides in chart 42-2. We should cover those.
We should. It's important to be aware. While they might enhance performance, the side effects can be severe and aren't listed impartially. Things like bad acne, shrinking of the testes, dramatically reduced or absent sperm count azoospermia,
And gynecomastia, breast development.
yes, because excess androgens can be converted to estrogens, specifically estradiol, in peripheral tissues like fat. This leads to breast enlargement in males. It disrupts the whole hormonal balance.
Okay, so what happens when that balance fails naturally? Let's talk about hypogonadism.
Hypogonadism just means reduced function of the gonads, specifically low testosterone production and a donivore low sperm count. The key is figuring out where the problem lies.
The source breaks it down into primary, secondary, and tertiary causes. Primary means the problem is in the tests themselves.
Correct. The testes aren't responding properly, maybe due to damage from infection like mumps or a genetic condition like Klinefelter syndrome. In this case, the pituitary tries to compensate.
So you'd see low testosterone, but high levels of LH and FSH because there's no negative feedback.
Exactly. The pituitary is shouting, but the tests aren't listening.
And secondary or tertiary hypogonadism.
That means the problem is upstream, either in the pituitary, secondary, or the hypothalamus, tertiary. They aren't sending out enough LH and FSH.
So in that case, you'd see low testosterone and low LH FSH.
Right. The tests are capable, but they aren't getting the signal to produce.
What are the typical symptoms in adults?
They can be varied fatigue, depression, decreased libido, erectile dysfunction, loss of muscle mass, decreased bone density. It's quite systemic.
And the source mentions a really important clinical link, especially between hypogonadism, ED, and heart disease.
Yes, this is a crucial connection. Low testosterone and erectile dysfunction, particularly when appearing together, are increasingly recognized as potential early warning signs for underlying cardiovascular disease, especially in men with type 2 diabetes.
Why is that? What's the common link?
It often comes down to endothelial dysfunction problems with the lining of blood vessels. The same processes like atherosclerosis or impaired nitric oxide production that affect the small arteries in the penis can also be affecting coronary arteries or other vessels.
So ED isn't just a quality of life issue. It can be a serious vascular red flag.
Absolutely. It warrants a closer look at cardiovascular risk factors.
Okay, let's shift gears to the mechanics of the sexual act itself. The source outlines 4 stages: erection, emission, ejaculation, detumescence. How is erection controlled?
Erection is primarily a parasympathetic nervous system event. Stimulation leads to the release of neurotransmitters that relax the smooth muscle in the arteries of the penis.
Allowing blood to flow in.
Exactly. Blood rushes into the erectile tissues, the corpora cavernosa and corpus spongiosum, causing them to engorge and become rigid.
And the key chemical mediator for that relaxation is nitric oxide.
Yes, nitric oxide, often referred to as a NANC, non-adrenergic, non-cholinergic mediator. It causes rapid vasodilation. Detumescence, or returning to the flaccid state, involves sympathetic signals causing vasoconstriction.
So parasympathetic for up, sympathetic for down, but then the sympathetic system takes over again later.
Right. For emission and ejaculation, control switches to the sympathetic nervous system, specifically nerves originating from the L1 and L2 spinal segments.
What happens during emission?
Emission is the movement of sperm from the epididymis and vas deferens into the prostatic urethra, where it mixes with fluids from the seminal vesicles and prostate. Critically, sympathetic signals also cause the internal urethral sphincter at the base of the bladder to close tightly.
Why is closing that sphincter so important?
To prevent retrograde ejaculation semen going backwards into the bladder instead of forwards out the urethra.
Okay, so emission loads the chamber, then ejaculation.
Ejaculation is the forceful expulsion of that semen. It involves rhythmic contractions of the bulbocavernosus and other pelvic floor muscles, along with contractions of the accessory glands themselves.
Got it. Finally, how does aging affect all this? The source suggests a gradual decline.
It's generally a slow, progressive decline in physiological efficiency, though fertility can certainly persist into older age.
Testosterone levels decrease.
Yes, there's a gradual drop, maybe starting around age 25 or 30, decreasing by roughly 10% per decade. This can contribute to symptoms sometimes vaguely termed andropause in older men, though it's not as distinct an event as menopause.
What are the physical changes?
The testes might become less firm and slightly smaller. Sperm production tends to decrease, though not necessarily stop. The prostate, as we discussed, usually enlarges. Ejaculatory force and volume often decrease.
And erectile dysfunction becomes more common too, often linked to those vascular issues we mentioned.
Very much so. Sclerotic changes, hardening of the arteries and veins within the penis, can impair the blood flow needed for a firm erection. It often reflects broader systemic vascular health.
Okay, let's try to pull this all together. We've covered a huge amount from the SRY gene determining sex.
And the crucial roles of AMH and testosterone, plus that DHT conversion for external structures.
Right. Then the need for careful temperature control via the dartos and pampiniform plexus.
Leading into the 90-day process of spermatogenesis in those incredibly long tubules supported by Sertoli cells.
The journey through the ducts, gating motility in the epididymis, storage in the ampulla.
And finally, mixing with the alkaline fluids from the seminal vesicles and especially the prostate, vital for neutralizing acidity.
Plus the whole HPG axis, GnRH, LH, FSH, testosterone, inhibit that elegant dual feedback loop.
And the switch from parasympathetic control for erection, mediated by nitric oxide, to sympathetic control for emission and ejaculation.
The big takeaway seems to be the intricate integration and balance required.
Absolutely. And how disruptions, whether it's hormonal, like in hypogonadism, or structural, like BPH, or vascular with aging and ED, can have widespread effects.
Yeah.
That link between ED, low T, and cardiovascular risk really highlights how interconnected things are. It's a robust system, but sensitive to imbalance.
So here's a final thought to chew on. Given that testosterone naturally declines with age, is this decline always a pathology to be treated, especially with testosterone replacement therapy becoming more common? Or is it perhaps the body finding a new, albeit different state of balance in later life? When should we step in?
That's the ongoing debate, isn't it? Weighing the potential benefits against the risks and understanding the body's own long-term adjustments. It's complex.
Indeed. Well, thank you for joining us on this deep dive through the source material. We hope breaking it down this way was helpful.
Hope so too. Until next time.